Stabilizing interactions. Van der Waals forces introduction. In molecular biology, the stability of biomolecules like proteins, nucleic acids, and lipids is governed by a variety of intermolecular interactions. These interactions are critical in determining the structure, function, and overall behavior of biological macromolecules. Among the many forces at play, van der Waals interactions are some of the weakest yet most ubiquitous stabilizing forces. They play a crucial role in stabilizing the three-dimensional structures of macromolecules and in mediating molecular recognition processes. 1. Understanding van der Waals forces. Van der Waals forces refer to the weak, short-range interactions that occur between molecules or atoms due to transient fluctuations in electron distribution. Despite their relative weakness compared to covalent and ionic bonds, these forces are fundamental in the stabilization of complex molecular structures and in facilitating interactions between biomolecules. There are three primary types of van der Waals interactions. A. London dispersion forces, induced dipole interactions. These are the weakest of the van der Waals forces. London dispersion forces arise due to instantaneous, momentary dipoles created by fluctuations in the electron density around an atom or molecule. These dipoles induce a corresponding dipole in neighboring molecules, leading to an attractive force between them. While these forces are present in all molecules, they are particularly significant in nonpolar molecules where dipole-dipole interactions do not occur. B. Dipole-dipole interactions, Keesom interactions. These interactions occur between molecules that have permanent dipoles, such as water. When the positive end of one polar molecule is close to the negative end of another, an attractive force is generated. This type of interaction is more significant than London dispersion forces because the dipoles in question are permanent, rather than transient. C. Dipole-induced dipole interactions, Debye interactions. In this type of interaction, a molecule with a permanent dipole induces a dipole in a neighboring nonpolar molecule. The interaction between the permanent dipole and the induced dipole results in an attractive force. This interaction is generally weaker than dipole-dipole interactions but still plays a role in stabilizing molecular interactions. 2. Role of van der Waals forces in biomolecular interactions. Despite their weak nature, van der Waals forces are integral to the overall stability of biomolecules, including proteins, nucleic acids, and lipids. A. Protein folding and stability. In proteins, van der Waals forces help stabilize the three-dimensional structure by promoting close packing of hydrophobic side chains in the interior of the protein. These forces contribute to the tertiary structure of proteins, ensuring that the protein adopts its functional conformation. The hydrophobic effect, driven in part by van der Waals interactions, plays a crucial role in the folding of proteins, where nonpolar amino acids aggregate in the core, away from the aqueous environment. B. Molecular recognition. Van der Waals interactions are involved in the recognition between molecules, such as in enzyme-substrate interactions or antibody-antigen binding. These interactions, although weak on their own, can become significant when there are many molecules participating in the interaction. The induced fit model of enzyme activity, for instance, suggests that slight changes in the enzyme or substrate structure allow for optimal van der Waals interactions at the binding site, which is essential for catalysis. C. Nucleic acid structure and function. In the case of nucleic acids, van der Waals forces play an essential role in stabilizing the double helix structure of DNA. The nitrogenous bases in DNA are stacked upon one another in the interior of the helix, and the interactions between these base pairs, primarily through van der Waals forces, help stabilize the helical structure. These interactions, combined with hydrogen bonding between complementary bases, contribute to the overall stability and function of the DNA molecule. D. Membrane stability and lipid bilayers. In biological membranes, lipid bilayers are stabilized by van der Waals forces between the hydrocarbon tails of phospholipids. These interactions help maintain the integrity of the membrane while allowing fluidity, which is crucial for membrane function. The van der Waals interactions between lipid molecules enable the membrane to maintain its structure while permitting lateral movement of lipids and proteins within the bilayer. 3. Van der Waals radius and steric effects. Each atom has an associated van der Waals radius, which is the distance at which the outer electron cloud of an atom can interact with neighboring atoms. The steric hindrance effect refers to the repulsion between atoms when they are too close to each other. The van der Waals radius plays a role in determining how closely two molecules can approach one another, as well as the strength of the interactions between them. A. Steric hindrance in molecular interactions. Steric hindrance can limit the efficiency of biochemical processes, such as enzyme catalysis or protein-protein interactions. For example, if two molecules approach too closely, their electron clouds may overlap, resulting in repulsive van der Waals forces that oppose the interaction. In such cases, molecules may need to undergo conformational changes to avoid steric clashes. 4. Van der Waals forces in drug design. The application of van der Waals interactions extends beyond natural systems and into fields such as pharmacology. In drug design, understanding and manipulating van der Waals interactions is crucial for the development of drugs that interact specifically with their targets, such as enzymes or receptors. A. Drug receptor binding. The binding of a drug to its receptor involves numerous intermolecular interactions, including hydrogen bonding, ionic interactions, and van der Waals forces. These interactions determine the affinity and selectivity of the drug for its receptor. By optimizing van der Waals interactions, drug designers can increase the binding strength and specificity of the drug, improving its efficacy and reducing side effects. B. Docking studies. Molecular docking simulations, used in drug discovery, rely on the principles of van der Waals forces, among other interactions, to predict how a drug molecule will fit into the binding site of its target protein. These studies help identify potential drug candidates and refine their structures to enhance their binding affinity. 5. Importance of van der Waals interactions in nature. Although van der Waals forces are relatively weak individually, their cumulative effect is profound in biological systems. These interactions contribute to the fine-tuning of molecular structures and the efficiency of biological processes. They are essential for the dynamic equilibrium that sustains life, allowing molecules to interact, fold, and function in a highly coordinated manner. Previous year solved question. Which of the following types of interactions is primarily responsible for stabilizing the helical structure of DNA? A. Ionic interactions. B. Hydrogen bonding. C. Van der Waals interactions. D. Disulfide bonds. Answer. Correct option, C. Van der Waals interactions. Explanation. The helical structure of DNA is primarily stabilized by the stacking of nitrogenous bases, which are held together by van der Waals interactions. These interactions contribute to the overall stability of the double helix, along with hydrogen bonding between complementary base pairs. Multiple choice questions, MCQs. 1. Which of the following is the primary characteristic of van der Waals interactions? A. Long-range interactions. B. Strong attractive forces. C. Short-range weak forces. D. Covalent bond formation. Answer. C. Short-range weak forces. 2. What type of van der Waals interaction occurs between two nonpolar molecules? A. Dipole-dipole interactions. B. London dispersion forces. C. Dipole-induced dipole interactions. D. Ionic interactions. Answer. B. London dispersion forces. 3. Which of the following is most likely to be stabilized by van der Waals forces in a protein? A. The primary sequence of amino acids. B. The secondary structure of the protein. C. The tertiary structure of the protein. D. The quaternary structure of the protein. Answer. C. The tertiary structure of the protein. This concludes the lecture on van der Waals forces. Let me know if you'd like to continue with another topic. You said, electrostatic. ChatGPT said. Electrostatic interactions in biomolecules. Introduction. Electrostatic interactions are one of the most important types of intermolecular forces in biological systems. These forces arise from the attraction or repulsion between charged particles, such as ions or polar molecules. In the context of biomolecules, electrostatic interactions play a central role in determining the structure, stability, and function of proteins, nucleic acids, lipids, and other biological macromolecules. These interactions can occur both within and between molecules, contributing to processes such as enzyme catalysis, molecular recognition, and DNA replication. 1. Understanding electrostatic interactions. Electrostatic forces result from the presence of electric charges on atoms or molecules. These charges can be either positive or negative, and the interactions between them are governed by the laws of electrostatics. The most notable principle governing electrostatic interactions is Coulomb's law, which describes the force between two charged objects. The strength of the electrostatic force F between two charged entities is directly proportional to the product of their charges Q1 and Q2, and inversely proportional to the square of the distance r between them. Mathematically, Coulomb's law is expressed as F equals K dot operator Q1 dot operator Q2 R2 F equals R2. K dot operator Q1 dot operator Q2. Where F is the electrostatic force, K is Coulomb's constant, Q1 and Q2. Q2 are the magnitudes of the charges, R is the distance between the charges. Types of electrostatic interactions. Electrostatic interactions can be categorized into two main types. A. Ion-ion interactions. These occur between two oppositely charged ions. The attraction between positively charged ions, cations, and negatively charged ions, anions, is a classic example of electrostatic interactions. In biological systems, ion-ion interactions are important for maintaining the stability of protein structures, for example, in salt bridges, which occur between oppositely charged side chains of amino acids and proteins. B. Dipole-dipole interactions. These occur between polar molecules, which have a permanent dipole moment. The partial positive charge of one molecule is attracted to the partial negative charge of another. In biomolecules, dipole-dipole interactions are crucial for the interaction between protein side chains and other polar molecules, such as water. These interactions are also important in the recognition of ligands by receptors and an enzyme substrate binding. 2. Electrostatic interactions in biomolecular structure. A. Role in protein structure. Electrostatic interactions are crucial for the stabilization of protein structures at various levels, including secondary, tertiary, and quaternary structures. Salt bridges. One of the most important examples of electrostatic interactions in proteins is the formation of salt bridges. Salt bridges occur when oppositely charged side chains of amino acids come into close proximity, forming an ionic bond. These interactions help stabilize the protein's tertiary structure, especially in the interior of globular proteins, where charged amino acid residues are often varied. Charge distribution. The distribution of charged residues on the surface of a protein can also affect its overall stability and function. The electrostatic surface potential of a protein influences its interaction with other molecules, such as substrates, inhibitors, or other proteins. B. Role in DNA and RNA structure. Electrostatic interactions are essential for maintaining the structure of nucleic acids like DNA and RNA. Phosphate backbone. In DNA, the phosphate groups in the sugar phosphate backbone are negatively charged. These charges create electrostatic repulsion between the strands of the double helix. To counteract this, positively charged ions, such as magnesium Mg² or sodium Na, are often present in the surrounding environment to neutralize the negative charges and stabilize the helical structure. Base pair stacking. In addition to hydrogen bonding, electrostatic interactions between the aromatic rings of nitrogenous bases also contribute to the stability of the double helix. The stacking of base pairs is influenced by both van der Waals forces and electrostatic interactions, which collectively stabilize the DNA structure. C. Electrostatic interactions in membrane proteins. In biological membranes, the electrostatic interactions between membrane proteins and lipid molecules are crucial for the function of membrane-bound enzymes, receptors, and channels. These interactions help maintain the integrity of the membrane and influence protein conformation and activity. The electrostatic interaction between charged regions of proteins and lipid head groups can help anchor the proteins in the membrane or facilitate their movement across the membrane. 3. Electrostatic interactions in molecular recognition. Electrostatic interactions are fundamental in the process of molecular recognition, where one biomolecule specifically interacts with another. This process is essential for a wide variety of biological phenomena, including A. Enzyme substrate binding. In enzyme catalysis, the binding of a substrate to an enzyme's active site is often facilitated by electrostatic interactions. The substrate may contain charged or polar groups that form favorable electrostatic interactions with the enzyme's active site, stabilizing the transition state and lowering the activation energy of the reaction. B. Antibody-antigen recognition. In the immune system, antibodies recognize specific antigens, foreign molecules, based on the electrostatic complementarity between their binding sites and the antigenic epitopes. This recognition is primarily driven by electrostatic interactions, along with hydrogen bonding and hydrophobic interactions, which help to form a stable complex between the antibody and the antigen. C. DNA-protein interactions. The interaction between DNA and DNA-binding proteins, such as transcription factors or histones, is largely mediated by electrostatic interactions. The negatively charged DNA interacts with positively charged residues on the protein, allowing the protein to bind to specific DNA sequences and regulate gene expression. 4. Electrostatic interactions in drug design. Electrostatic interactions play a significant role in drug design and in the development of therapeutics that specifically target biological macromolecules. Understanding these interactions is critical for optimizing the binding of a drug to its target and improving the selectivity and efficacy of the drug. A. Target specificity. By modulating the electrostatic complementarity between a drug molecule and its target protein, drug designers can increase the binding affinity and specificity of the drug. Drugs can be designed to exploit specific electrostatic interactions at the active site of an enzyme or receptor, improving their therapeutic effect. B. Protein-protein interactions. Electrostatic interactions also play a role in protein-protein interactions, which are important in regulating cellular processes such as signal transduction. Inhibitors that disrupt electrostatic interactions between proteins can be developed to interfere with signaling pathways in diseases like cancer. 5. Previous year solved question. Which of the following interactions are primarily responsible for the binding of an enzyme to its substrate? A. Hydrogen bonds. B. Electrostatic interactions. C. Hydrophobic interactions. D. Covalent bonds. Answer. Correct option, B. Electrostatic interactions. Explanation. The binding of an enzyme to its substrate is primarily mediated by electrostatic interactions between charged or polar groups in the enzyme's active site in the substrate. While hydrogen bonds and hydrophobic interactions also contribute, electrostatic interactions play a central role in stabilizing the enzyme substrate complex. Multiple choice questions. MCQs. 1. Which of the following interactions is most responsible for stabilizing the tertiary structure of proteins? A. Hydrogen bonding. B. Electrostatic interactions. C. Covalent bonds. D. Van der Waals interactions. Answer. B. Electrostatic interactions. 2. The interaction between the negatively charged phosphate backbone of DNA and positively charged ions in the surrounding environment is an example of a dipole-dipole interactions. B. Ion-dipole interactions. C. Ion-ion interactions. D. Hydrophobic interactions. Answer. C. Ion-ion interactions. 3. Electrostatic interactions play an important role in a. Stabilizing protein secondary structure. B. The formation of salt bridges in proteins. C. Protein DNA interactions. D. All of the above. Answer. D. All of the above. This concludes the lecture on electrostatic interactions. Feel free to ask for more topics or any clarifications. You said. Hydrogen bonding, hydrophobic interaction. ChatGPT said. Hydrogen bonding and hydrophobic interactions in biomolecules. Introduction. In biological systems, the structure and function of macromolecules like proteins, nucleic acids, and lipids are heavily influenced by various types of intermolecular interactions. Among these, hydrogen bonding and hydrophobic interactions are two of the most significant forces that contribute to the stability, conformation, and functionality of biological molecules. These interactions, though distinct in their nature, work in concert to regulate the shape and activity of biomolecules, influencing processes such as protein folding, DNA stability, enzyme catalysis, and molecular recognition. 1. Hydrogen bonding in biomolecules. A. Definition and basic principles. A hydrogen bond is a weak to moderate attractive force that exists between a hydrogen atom covalently bonded to an electronegative atom, usually nitrogen, oxygen, or fluorine, and another electronegative atom with a lone pair of electrons. The key feature of hydrogen bonds is the polar covalent bond, where the hydrogen atom has a partial positive charge, delta plus, due to its bond with a highly electronegative atom. The electronegative atom to which it is bonded, such as oxygen or nitrogen, carries a partial negative charge, delta minus, making it capable of forming a hydrogen bond with another electronegative atom. Mathematically, the strength of a hydrogen bond is much weaker than a covalent bond but stronger than typical van der Waals interactions. Hydrogen bonds are directional, meaning they are strongest when the donor, hydrogen, and acceptor, electronegative atom, are aligned. B. Hydrogen bonds in protein structure. In proteins, hydrogen bonds are crucial for maintaining the secondary, tertiary, and quaternary structures. They help stabilize structures such as alpha helices and beta sheets. Alpha helix. In an alpha helix, the backbone amide nitrogen of one amino acid forms a hydrogen bond with the backbone carbonyl oxygen of an amino acid that is typically 4 residues away in the chain. These bonds contribute significantly to the helical structure and its stability. Beta sheets. In beta sheets, hydrogen bonds form between the backbone amide and carbonyl groups of adjacent strands. These bonds are responsible for the sheet-like arrangement of the polypeptide chain, which contributes to the overall structural integrity of proteins. C. Hydrogen bonds in nucleic acids. In DNA, hydrogen bonds are the key forces that hold together the two complementary strands of the double helix. Base pairs in DNA, adenine, thymine, and guanine, cytosine, are held together by hydrogen bonds. AT pairs form 2 hydrogen bonds, while GC pairs form 3 hydrogen bonds, contributing to the stability and specificity of base pairing in the double-stranded structure. In RNA, hydrogen bonds are also essential for stabilizing the secondary structure, such as the formation of hairpins and loops, and for the proper function of ribosomal RNA in translation. 2. Hydrophobic interactions in biomolecules. A. Definition and basic principles. Hydrophobic interactions refer to the tendency of nonpolar molecules or nonpolar parts of molecules to aggregate together in an aqueous environment, away from the polar solvent water. This interaction is driven by the exclusion of water molecules from nonpolar surfaces, rather than a direct attraction between nonpolar molecules. In aqueous solutions, water molecules form an ordered structure around nonpolar molecules, leading to an increase in free energy. The aggregation of nonpolar groups reduces this ordering and decreases the overall free energy, stabilizing the system. Although hydrophobic interactions are technically not bonds, they are a fundamental force in maintaining the structure of biological molecules. B. Hydrophobic interactions in proteins. Hydrophobic interactions play a pivotal role in protein folding. Hydrophobic amino acid residues tend to cluster in the interior of a protein, away from the aqueous environment, while hydrophilic polar residues are often found on the exterior. This segregation helps drive the folding of proteins into their most stable conformations, a process known as hydrophobic collapse. The exposure of hydrophobic side chains to water would result in an unfavorable increase in free energy, while their burial inside the protein structure is energetically favorable. C. Hydrophobic interactions in membranes. Hydrophobic interactions are also critical in the formation and stability of biological membranes. The hydrophobic tails of phospholipids aggregate together in the interior of the bilayer, while the hydrophilic heads interact with the aqueous environment. This arrangement forms a stable membrane structure that acts as a selective barrier, allowing cells to maintain distinct internal and external environments. D. Hydrophobic interactions in ligand binding. Hydrophobic interactions are involved in molecular recognition, particularly in the binding of ligands to their target proteins. Nonpolar regions of ligands and receptors often form hydrophobic interactions, contributing to the specificity and strength of the binding. 3. Hydrogen bonding and hydrophobic interactions in molecular function. A. Enzyme catalysis. Both hydrogen bonding and hydrophobic interactions are vital in the binding of substrates to enzymes. Hydrogen bonds help orient the substrate properly in the enzyme's active site, facilitating the formation of the transition state. Meanwhile, hydrophobic interactions contribute to the stability of the enzyme-substrate complex and also play a role in the enzyme specificity for particular substrates. B. DNA replication and transcription. Hydrogen bonding is essential for the complementary base pairing in DNA replication and transcription. In DNA replication, the enzyme DNA polymerase facilitates the formation of hydrogen bonds between complementary bases on the template strand and the incoming nucleotide, ensuring accurate replication of genetic material. Similarly, in RNA transcription, hydrogen bonds between RNA nucleotides and the DNA template allow the synthesis of an RNA strand complementary to the DNA template. C. Protein-protein interactions. Both hydrogen bonds and hydrophobic interactions contribute to protein-protein interactions. Hydrophobic residues often mediate the association between proteins, while hydrogen bonds can further stabilize the protein-protein complex. For example, in the formation of protein complexes like the ribosome, hydrogen bonds help maintain the alignment of protein subunits, while hydrophobic interactions help the subunits come together. 4. Previous year solved question. Which of the following is the primary driving force for the folding of a protein into its native three-dimensional structure? A. Hydrogen bonding. B. Hydrophobic interactions. C. Disulfide bonds. D. Electrostatic interactions. Answer. Correct option, B. Hydrophobic interactions. Explanation. While hydrogen bonding, disulfide bonds, and electrostatic interactions contribute to protein structure, the primary driving force for protein folding is hydrophobic interactions. The hydrophobic residues tend to fold into the interior of the protein, avoiding exposure to water, thus stabilizing the folded structure. Multiple choice questions. MCQs. 1. Which of the following is primarily responsible for the stable pairing of bases in the double helix structure of DNA? A. Hydrogen bonding. B. Hydrophobic interactions. C. Covalent bonds. D. Electrostatic interactions. Answer. A. Hydrogen bonding. 2. The formation of alpha helices in proteins is stabilized by A. Hydrophobic interactions. B. Hydrogen bonds. C. Ionic bonds. D. Van der Waals forces. Answer. B. Hydrogen bonds. 3. In the context of protein folding, the burial of hydrophobic amino acids in the interior of the protein is an example of A. Hydrogen bonding. B. Hydrophobic interactions. C. Ionic bonds. D. Covalent bonds. Answer. B. Hydrophobic interactions. This concludes the lecture on hydrogen bonding and hydrophobic interactions. Feel free to ask for any further topics or clarifications. You said. Conformation of proteins. Ramachandran plot. Secondary structure. ChatGPT said. Conformation of proteins. Ramachandran plot and secondary structure. Introduction. The three-dimensional structure of a protein is essential for its function. Understanding how a protein achieves its specific conformation is key to comprehending how it performs its biological roles. Protein conformation is determined by the sequence of amino acids and the interactions among them, leading to the folding of the polypeptide chain into a specific 3D shape. This process is guided by several structural motifs, including the secondary structure elements, alpha helices, beta sheets, as well as tertiary and quaternary structures. A critical tool for visualizing and understanding the conformational constraints of proteins is the Ramachandran plot, which illustrates the steric limitations of protein backbone dihedral angles and provides insight into the possible secondary structure formations in a protein. 1. Ramachandran plot. A. Overview and definition. The Ramachandran plot, introduced by G. N. Ramachandran and co-workers in 1963, is a graphical representation that plots the dihedral angles of the protein backbone, the phi and psi angles. These angles represent the rotation around the bonds in the protein's backbone chain. Phi, the angle of rotation about the bond between the nitrogen and alpha carbon, and C alpha, psi, the angle of rotation about the bond between the alpha carbon and carbonyl carbon, C alpha C. The Ramachandran plot maps these two angles for all the amino acid residues in a given protein and displays them in a way that shows the regions where certain conformations are sterically allowed, and others are disallowed due to physical constraints. B. The importance of the Ramachandran plot. The Ramachandran plot is invaluable in protein structure analysis because it helps identify the sterically favorable and unfavorable regions of the polypeptide backbone. The primary significance of the plot is in confirming the quality and validity of a protein's three-dimensional structure. Allowed regions. These correspond to specific conformations where the steric clashes are minimal, allowing the backbone angles to assume stable configurations, typically forming regular secondary structures like alpha helices and beta sheets. Disallowed regions. These correspond to conformations where the dihedral angles would cause atomic clashes, resulting in sterically forbidden configurations. The plot is often divided into 4 regions. Alpha helix, characterized by specific phi and psi angles, typically around -60 degrees and -45 degrees. Beta sheet, characterized by phi and psi angles near -120 degrees and +120 degrees. Left-handed helix, a rare conformation where phi and psi angles fall at values around +60 degrees and +60 degrees. Other allowed regions, representing various turns, loops, and irregular regions in protein structure. The Ramachandran plot helps to visualize the protein's folding, ensuring that the structure adopted by a protein is energetically stable and feasible, and serves as a tool for validating experimental structures derived from X-ray crystallography or NMR spectroscopy. C. Analysis of the plot. In the context of protein modeling, the Ramachandran plot can be used to assess the quality of a protein structure. A well-folded protein should show the majority of its residues in the most favored regions of the plot. A protein with unfavored regions might suggest errors in its predicted structure, prompting further refinement or reconsideration of the model. For example, the ideal alpha helix region falls at phi approximately equals -60 degrees and psi approximately equals -45 degrees, and the beta sheet region at phi approximately equals -120 degrees and psi approximately equals +120 degrees. 2. Secondary structure of proteins. A. Definition of secondary structure. The secondary structure of proteins refers to local folding patterns that arise from the interactions between the backbone atoms in the polypeptide chain. These interactions, mainly hydrogen bonds, determine how the protein chain bends and folds at a local level, and they form regular structural motifs such as alpha helices, alpha helix, beta sheets, beta turns, random coils or loops. B. Alpha helix. The alpha helix is one of the most common and stable secondary structures found in proteins. It is a right-handed coiled structure stabilized by hydrogen bonds between the backbone amide hydrogen of one residue and the carbonyl oxygen of a residue 4 positions earlier in the sequence. This regular, helical arrangement allows the protein chain to take on a compact and stable conformation. Each turn of the alpha helix typically contains 3.6 amino acid residues, and the distance between two adjacent turns is about 0.54 nanometers, 5.4 A. The alpha helix is often found in membrane proteins and structural proteins, where its rigidity and stability are essential for maintaining the structural integrity of the protein. C. Beta sheets. The beta sheet is another major type of secondary structure, formed when two or more polypeptide chains or strands align in a parallel or anti-parallel fashion. These strands are connected by hydrogen bonds between the carbonyl oxygen of one strand and the amide hydrogen of an adjacent strand. The beta sheet can be parallel, the strands run in the same direction; anti-parallel, the strands run in opposite directions. Anti-parallel beta sheets are generally more stable due to the optimal alignment of hydrogen bonds. The beta turns, often found between the strands of a beta sheet, enable the polypeptide chain to reverse direction and fold compactly. D. Beta turns and loops. Beta turns are short, sharp turns in the protein backbone that allow the polypeptide chain to change direction. These are typically composed of 4 amino acid residues and are stabilized by hydrogen bonds between the first and fourth residues. Turns are crucial for the formation of compact protein structures and for maintaining the overall folding of the protein. Loops, unlike regular secondary structures like alpha helices or beta sheets, do not follow a repetitive pattern. They often connect different secondary structural elements and contribute to the flexibility and functionality of the protein. Loops are particularly important in the active sites of enzymes and in regions involved in protein-protein interactions. 3. Tertiary and quaternary structures. While secondary structures like alpha helices and beta sheets form the local backbone folding patterns of a protein, the tertiary structure describes the overall 3D shape formed by the complete folding of the polypeptide chain. This is influenced by various interactions, including hydrogen bonding, hydrophobic interactions, ionic bonds, and disulfide bridges. The quaternary structure refers to the arrangement of multiple polypeptide subunits, if present in a multi-subunit protein. Quaternary structure is stabilized by the same interactions that stabilize tertiary structure, but occurs between different polypeptide chains. 4. Previous year solved question. Which of the following secondary structure motifs is most commonly associated with the formation of the DNA binding domain of proteins? A. Alpha helix. B. Beta sheet. C. Beta turns. D. Random coil. Answer. Correct option, A. Alpha helix. Explanation. The alpha helix is commonly found in the DNA binding domains of proteins. This structural motif is well suited for forming stable interactions with the negatively charged backbone of DNA. The alpha helix is often referred to as a helix-turn helix motif, which is involved in DNA recognition and binding. Multiple choice questions. MCQs 1. The Ramachandran plot primarily helps to determine a tertiary structure of proteins. B. Stability of a protein's quaternary structure. C. Sterically allowed regions of the protein backbone. D. Sequence of amino acids in a protein. Answer. C. Sterically allowed regions of the protein backbone. 2. Which of the following secondary structures is stabilized by hydrogen bonds between the carbonyl oxygen of one strand and the amide nitrogen of an adjacent strand? A. Alpha helix. B. Beta sheet. C. Random coil. D. Beta turn. Answer. B. Beta sheet. 3. Which of the following is the most common secondary structure in proteins involved in enzyme catalysis? A. Beta turns. B. Beta sheets. C. Random coils. D. Alpha helices. Answer. D. Alpha helices. This concludes the lecture on conformation of proteins, covering Ramachandran plots and secondary structure. If you have any further questions or need additional explanations, feel free to ask. You said. Domains, motif and folds. ChatGPT said. Domains, motifs, and folds in proteins. Introduction. Proteins are complex macromolecules that perform a vast array of functions in the cell, ranging from catalysis of biochemical reactions to structural support and regulation. These diverse functions are largely dependent on the protein's three-dimensional structure. Proteins are composed of linear chains of amino acids, but their final functional form arises from the folding of these chains into specific structural patterns. Understanding the concepts of domains, motifs, and folds is crucial in protein structure-function relationships, as these structural elements dictate how proteins interact with other molecules and carry out their biological roles. 1. Domains. A. Definition. A domain in a protein refers to a distinct, compact, and functional region of the protein structure that can fold independently of the rest of the molecule. A domain is typically a modular unit that has its own unique structural and functional properties. Domains often correspond to specific functional units within a protein, and many proteins consist of multiple domains, each responsible for a different aspect of the protein's activity. B. Characteristics of protein domains. Autonomous folding. Domains can fold independently of other parts of the protein. This allows them to maintain stability and functionality even when isolated. Functional specialization. Each domain often corresponds to a specific function. For example, an enzyme may contain a catalytic domain, a substrate binding domain, and a regulatory domain. Modularity. Domains can be combined in various ways to form multi-domain proteins, which may have complex functionalities derived from the interaction of their domains. Conservation. Domains are often evolutionarily conserved across different species and even across different proteins that perform similar functions. This conservation is essential for the maintenance of protein functionality across evolutionary time. C. Examples of protein domains. SH2-SRC homology 2 domain. This domain recognizes and binds to phosphorylated tyrosine residues, playing a critical role in signal transduction. Kinase domain. Found in protein kinases, this domain catalyzes the transfer of phosphate groups from ATP to proteins. DNA binding domain. Present in many transcription factors and enzymes, this domain interacts with specific sequences in the DNA, allowing proteins to regulate gene expression. Domains can be thought of as the building blocks of protein structure, as they are frequently reused in different proteins. 2. Motifs. A. Definition. A motif is a short, conserved sequence or pattern of secondary structural elements, e.g., alpha helices and beta sheets, that forms a small part of a larger protein structure. Unlike domains, motifs do not fold independently and may require other parts of the protein to stabilize them. A motif often contributes to the overall function of the protein, particularly in interactions with other molecules. B. Characteristics of protein motifs. Smaller than domains, motifs are typically smaller regions of a protein compared to domains. They usually consist of a few amino acids or a short segment of secondary structure. Functional significance. Motifs are often associated with specific biological functions, such as enzyme catalysis, molecular recognition, or protein-protein interactions. Structural stability. While motifs are stable in the context of a full protein, they do not necessarily possess the stability or independence of a domain. A motif is often stabilized by the larger protein context. C. Examples of protein motifs. Zinc. The larger protein context. C. Examples of protein motifs. Zinc finger motif, a common motif found in DNA-binding proteins. It consists of a beta sheet and an alpha helix that coordinates the zinc ion, allowing the protein to interact with DNA. Helix-turn helix HTH motif, a structural motif that plays a key role in DNA binding. It consists of two alpha helices connected by a short turn, allowing the protein to insert into the major groove of the DNA helix. Leucine zipper motif. This motif consists of repeated leucine residues spaced every 7 amino acids and is involved in the formation of protein dimers, particularly in transcription factors. Motifs are essential for protein function, as they often provide the structural framework for molecular recognition or interaction with other biomolecules. 3. Folds. A. Definition. A fold refers to the overall three-dimensional arrangement of a protein's secondary structure elements, such as alpha helices and beta sheets, into a stable and functional conformation. The fold of a protein is dictated by the amino acid sequence and the interactions between its constituent atoms, and it determines the protein's functional properties. Protein folds are classified into different categories based on the general arrangement of secondary structural elements. B. Characteristics of protein folds. Stability. The fold of a protein is stabilized by various interactions, including hydrogen bonds, hydrophobic interactions, and electrostatic interactions. Conservation. Like domains, protein folds are often conserved across different proteins. Proteins that share the same fold usually have similar functions, even if they are involved in different biological processes. Functionality. The fold of a protein is crucial to its function. A protein's active site, for example, is often formed by the specific arrangement of its secondary structure elements in the folded conformation. See examples of protein folds. Globular fold. Many enzymes and other functional proteins have a globular fold, which is a compact, spherical arrangement of secondary structures. The fold allows for efficient packing of the protein and is often seen in proteins involved in metabolism and signaling. Alpha-beta fold. This fold consists of alternating alpha helices and beta sheets. It is commonly found in enzymes that catalyze complex biochemical reactions, such as hydrolases and oxidoreductases. Beta barrel. A fold formed by beta sheets that are arranged in a barrel-like structure. This fold is often found in membrane proteins, such as porins, which form channels across membranes. 4. Relationship between domains, motifs, and folds. Domains are the largest and most independent structural units in a protein, often associated with specific functions. They may contain several motifs and contribute to the overall fold of the protein. Motifs are smaller, often recurring structural elements that play a role in the protein's function. They contribute to specific interactions within a domain or between domains. Folds are the overall three-dimensional structure of the protein, defined by the arrangement of secondary structural elements, alpha helices, beta sheets, into stable and functional shapes. The fold of a protein dictates how its domains are arranged, and the motifs within those domains determine specific aspects of function. Understanding the interplay between these structural elements is crucial for comprehending how proteins work. 5. Previous year solved question. Which of the following is a characteristic of protein domains? A. Domains are always composed of a single type of secondary structure. B. A domain can fold independently of the rest of the protein. C. A motif refers to the overall three-dimensional structure of a protein. D. Domains are not functionally relevant to proteins. Answer. Correct option. B. A domain can fold independently of the rest of the protein. Explanation. Domains are distinct, functional regions of proteins that can fold independently of the rest of the protein. This modularity is a key feature of domains, allowing them to be rearranged and combined in different proteins to perform various functions. Multiple choice questions. MCQs. 1. Which of the following motifs is commonly found in proteins that bind DNA? A. Zinc finger motif. B. Helix turn helix motif. C. Leucine zipper motif. D. All of the above. Answer. D. All of the above. 2. The overall 3D shape of a protein is referred to as its a primary structure. B. Secondary structure. C. Tertiary structure. D. Quaternary structure. Answer. C. Tertiary structure. 3. What is the characteristic feature of the beta barrel protein fold? A. Alpha helices are arranged in a parallel fashion. B. Beta. Answer. Correct option. 5. Previous year solved question. Which of the following is a characteristic of protein domains? A. Domains are always composed of a single type of secondary structure. B. A domain can fold independently of the rest of the protein. C. A motif refers to the overall three-dimensional structure of a protein. D. Domains are not functionally relevant to proteins. Answer. Correct option. B. A domain can fold independently of the rest of the protein. Explanation. Domains are distinct, functional regions of proteins that can fold independently of the rest of the protein. B. A domain can fold independently. Which of the following is a characteristic of protein domains? A. Domains are always composed of a single type of secondary structure. B. A domain can fold independently of the rest of the protein. C. A motif refers to the overall three-dimensional structure of a protein. D. Domains are not functionally relevant to proteins. Answer. Correct option. B. A domain can fold independently of the rest of the protein. Explanation. Domains are distinct, functional regions of proteins that can fold independently of the rest of the protein. This modularity is a key feature of domains, allowing them to be rearranged and combined in different proteins to perform various functions. Multiple choice questions. MCQs. 1. Which of the following motifs is commonly found in proteins that bind DNA? A. Zinc finger motif. B. Helix turn helix motif. C. Leucine zipper motif. D. All of the above. Answer. D. All of the above. 2. The overall 3D shape of a protein is referred to as its a primary structure. B. Secondary structure. C. Tertiary structure. D. Quaternary structure. Answer. C. Tertiary structure. 3. What is the characteristic feature of the beta barrel protein fold? A. Alpha helices are arranged in a parallel fashion. B. Beta sheets form a barrel-like structure. C. It is a rare motif found in membrane proteins. D. It contains a random coil structure. Answer. B. Beta sheets form a barrel-like structure. This concludes the lecture on domains, motifs, and folds. These structural elements are foundational to understanding protein function. Should you need further clarification or more practice questions, feel free to ask. You said. Conformation of nucleic acids helix A, B, Z, tRNA, microRNA. ChatGPT said. Conformation of nucleic acids helix types and RNA structures introduction. Nucleic acids, DNA and RNA, are the fundamental molecules that store and transmit genetic information in living organisms. The conformational structures of nucleic acids are critical to their function in processes like replication, transcription, translation, and regulation. The three-dimensional shape or conformation of nucleic acids is not only important for their stability but also for their interactions with other molecules. Among the various structural forms of nucleic acids, the helix forms of DNA A, B, and Z and the distinct structures of tRNA and microRNA play vital roles in genetic processes and cellular mechanisms. Understanding these conformations helps us comprehend their biological functions and interactions at a molecular level. 1. Helical forms of DNA A, B, Z, A. bDNA, right-handed double helix. Structure. bDNA is the most common and biologically significant form of DNA found under physiological conditions. It has a right-handed helix structure, with two antiparallel polynucleotide strands twisted around each other. Each strand consists of alternating sugar, deoxyribose, and phosphate groups, forming the backbone, with nitrogenous bases adenine, thymine, cytosine, and guanine projecting inward, forming base pairs. A pairs with T, and C pairs with G characteristics. Helical turn. There are about 10.5 base pairs per full turn of the helix. Major and minor grooves. bDNA has both major and minor grooves, which are important for protein binding, such as transcription factors and enzymes involved in DNA replication. Stability. bDNA is stabilized by hydrogen bonding between base pairs and hydrophobic interactions between stacked bases. B. aDNA, right-handed helix, dehydrated form. Structure. aDNA is a right-handed helix but has a more compact structure. bDNA, right. 1. Helical forms of DNA A, B, Z, A. bDNA, right-handed double helix. Structure. bDNA is the most common and biologically significant form of DNA found under physiological conditions. It has a right-handed helix structure, with two antiparallel polynucleotide strands twisted around each other. Each strand consists of alternating sugar deoxyribose and phosphate groups, forming the backbone, with nitrogenous bases adenine, thymine, cytosine, and guanine projecting inward, forming base pairs. A pairs with T and C pairs with G characteristics. Helical turn. There are about 10.5 base pairs per full turn of the helix. Major and minor grooves. bDNA has both major and minor grooves, which are important for protein binding, such as transcription factors and enzymes involved in DNA replication. Stability. bDNA is stabilized by hydrogen bonding between base pairs and hydrophobic interactions between stacked bases. aDNA, right-handed helix, dehydrated form. Structure. aDNA is a right-handed helix but has a more compact structure compared to bDNA. It forms under conditions of low humidity or high salt concentrations. In aDNA, the helix is more tightly wound with 11 base pairs per turn, and the sugar-phosphate backbone is tilted relative to the base pairs. Structure. aDNA is a right-handed helix but has a more compact structure compared to bDNA. It forms under conditions of low humidity or high salt concentrations. In aDNA, the helix is more tightly wound with 11 base pairs per turn, and the sugar-phosphate backbone is tilted relative to the base pairs. Characteristics. Helical turn, 11 base pairs per turn, making it more compact than bDNA. Base pair orientation. The base pairs are tilted relative to the helix axis and are not perpendicular, unlike in bDNA. Biological relevance. aDNA is rarely found in nature but can form transiently in some biological processes, especially when DNA is bound by certain proteins or exposed to high ionic conditions. zDNA, left-handed helix. Structure. zDNA is a left-handed helix, which is quite different from the right-handed helices of aDNA and bDNA. It forms when certain DNA sequences, particularly those with alternating purine pyrimidine repeats, such as GC-rich sequences, are present. The zDNA structure is characterized by a zigzag backbone with 12 base pairs per turn. Characteristics. Helical turn, 12 base pairs per turn. Right to left-handed transition. zDNA can form from bDNA under supercoiling or high ionic conditions and is believed to play a role in gene expression regulation and DNA packaging. Biological relevance. Though not common in typical DNA structures, zDNA may be involved in biological processes such as transcription and recombination, as well as regulation of gene activity. 2. tRNA, transfer RNA. A. Structure and function. Conformation. tRNA molecules are crucial for translating the genetic code into proteins. The tRNA molecule adopts a cloverleaf-like secondary structure, which contains four main stems, the acceptor stem, the D loop, the anticodon loop, and the T loop. The tertiary structure of tRNA results in an L shape, which is necessary for proper interaction with ribosomes during protein synthesis. Acceptor stem. This is where the amino acid attaches to the tRNA. The 3' end of the tRNA has a single nucleotide sequence, CCA, where the amino acid is esterified. Anticodon loop. This loop contains a 3-nucleotide sequence anticodon that pairs with the codon on the mRNA, ensuring the correct amino acid is added during translation. D loop and T loop. These loops are involved in stabilizing the overall structure and interactions with other molecules, including ribosomes and aminoacyl tRNA synthetase. Function. The primary function of tRNA is to transport specific amino acids to the ribosome during protein synthesis. The ternus anticodon recognizes the corresponding codon on mRNA, ensuring the correct amino acid is added to the growing polypeptide chain. 3. MicroRNA, mRNA. A. Structure and function. Conformation. MicroRNAs are small, non-coding RNA molecules, about 21 to 25 nucleotides long, that play a crucial role in regulating gene expression. mRNAs often fold into a hairpin-like structure, which is processed by the enzyme dicer into a mature, functional form. The structure consists of a stem double-stranded region and a looped single-stranded region. Mechanism. The mature mRNA guides the RNA-induced silencing complex RISC to target mRNA molecules. The mRNA base pairs with complementary sequences in the 3' untranslated region UTR of target mRNAs, leading to either degradation of the mRNA or inhibition of translation, thus regulating gene expression post-transcriptionally. Function. mRNAs are involved in various biological processes, including development, differentiation, apoptosis, and immune response. They help fine-tune gene expression by post-transcriptionally modulating the stability and translation of target mRNAs. 4. Relationship between the structures. DNA helix types A, B, Z. The different conformations of DNA A, B, and Z allow the molecule to adopt various shapes based on environmental conditions, providing flexibility for various biological processes, such as transcription and replication. The stability of these forms is critical for maintaining the integrity of genetic information and for interactions with regulatory proteins. tRNA. The unique L shape of tRNA is essential for its function and translation, as it ensures proper interaction with ribosomes and mRNA during protein synthesis. mRNA. The hairpin structure of mRNA is crucial for its function and gene regulation. The ability of mRNA to form double-stranded regions allows it to be processed and guide RISC to target mRNAs, effectively controlling gene expression. 5. Previous year solved question. Question. Which of the following best describes the structure of tRNA? A. tRNA adopts a cloverleaf-like structure and is involved in gene transcription. B. tRNA folds into a hairpin-like structure and is involved in protein translation. C. tRNA is a double-stranded RNA molecule with no specific secondary structure. D. tRNA consists of a single strand of nucleotides involved in DNA replication. Answer. Correct option. B. tRNA folds into a hairpin-like structure and is involved in protein translation. Explanation. tRNA is a small RNA molecule that adopts a cloverleaf-like secondary structure, forming an L shape in its tertiary conformation. This structure is essential for its function in protein translation, as it helps align amino acids with the correct codons on mRNA during ribosomal translation. Multiple choice questions MCQs. 1. Which form of DNA is most commonly found under physiological conditions? A. aDNA. B. bDNA. C. zDNA. D. None of the above. Answer. B. bDNA. 2. What is the primary function of tRNA in protein synthesis? A. To transport amino acids to the ribosome for protein assembly. B. To transcribe the genetic code from DNA to RNA. C. To catalyze the formation of peptide bonds in the ribosome. D. To bind to ribosomal RNA during translation. Answer. A. To transport amino acids to the ribosome for protein assembly. 3. What type of RNA plays a key role in post-transcriptional regulation of gene expression by binding to mRNA? A. tRNA. B. mRNA. C. rRNA. D. mRNA. Answer. D. mRNA. This concludes the lecture on the conformation of nucleic acids. These structural forms are vital for understanding nucleic acid function and interaction in cellular processes. Should you require more details or further practice, feel free to ask. You said. Acids, bases, and buffers, weak acids and weak bases, ChatGPT said. Acids, bases, and buffers, weak acids and weak bases. Introduction. Acids, bases, and buffers are fundamental concepts in biochemistry, playing essential roles in maintaining the pH and chemical environment of biological systems. They are involved in a variety of physiological processes, including enzyme activity, cellular respiration, and the regulation of blood pH. While strong acids and bases completely dissociate in aqueous solutions, weak acids and bases only partially dissociate. Buffers, which are typically composed of a weak acid and its conjugate base, or a weak base and its conjugate acid, help maintain the pH of a system by resisting changes in pH when small amounts of acid or base are added. 1. Weak acids. A. Definition and characteristics. Weak acids are acids that do not completely dissociate in water. They only partially ionize, meaning they release a small proportion of hydrogen ions H into the solution. This incomplete dissociation results in a reversible equilibrium between the acid HA, its dissociated ions H and A, and water. General reaction of weak acid dissociation, H. A rightwards harpoon over leftwards harpoon H plus A minus. Ha rightwards harpoon over leftwards harpoon H plus A minus. Ionization constant Ka. The dissociation of weak acids is characterized by the acid dissociation constant Ka, which is a measure of the strength of the acid. A small Ka value indicates a weak acid, meaning it dissociates less in solution. Ka equals H plus A minus HA. Ka equals Ha H plus A minus. Example of weak acids. Acetic acid CHCOOH in water. Acetic acid partially dissociates into acetate ions CHCOO and hydrogen ions H, establishing an equilibrium. Formic acid HCOOH. Similarly, formic acid dissociates slightly to release H and formate ions HCOO. B. pH of weak acids. The pH of a solution containing a weak acid depends on its concentration and the value of Ka. For weak acids, the concentration of H ions is determined using the equilibrium constant expression. Weak acids do not significantly lower the pH of a solution as strong acids do, since they release fewer H ions. 2. Weak bases. A. Definition and characteristics. Weak bases are bases that do not completely dissociate in water. When dissolved, weak bases only partially accept protons H or release hydroxide ions O into solution, resulting in an equilibrium between the base B and its conjugate acid BH. General reaction of weak base dissociation, B. Plus H2O rightwards harpoon over leftwards harpoon BH plus OH minus B plus H2. O rightwards harpoon over leftwards harpoon BH plus O minus. Base dissociation constant Kb. The strength of a weak base is measured by its base dissociation constant Kb, which reflects the extent of its proton accepting ability. A small Kb indicates a weak base. Kb equals BH plus OH minus B. Kb equals BH plus O minus. Example of weak bases. Ammonia NH. Ammonia is a weak base that reacts with water to form ammonium ions NH and hydroxide ions O. Methylamine CHNH. Similarly, methylamine accepts a proton from water, forming methylammonium ions CHNH and O. B. pH of weak bases. The pH of a solution containing a weak base is determined by the concentration of O ions it produces. Weak bases do not significantly raise the pH of a solution as much as strong bases do, since they release fewer O ions. 3. Buffers. A. Definition and role of buffers. Buffers are solutions that resist changes in pH when small amounts of acid or base are added. Buffers typically consist of a weak acid and its conjugate base or a weak base and its conjugate acid. They help maintain the pH of biological systems within a narrow range, which is essential for processes like enzyme activity and cellular function. B. Buffer capacity. The buffer capacity refers to the ability of a buffer solution to resist changes in pH. It is highest when the pH is close to the pKa of the weak acid or base in the buffer. Buffer capacity depends on the concentration of the acid-base pair. Higher concentrations provide greater resistance to pH changes. C. Henderson-Hasselbalch equation. The effectiveness of a buffer can be quantified using the Henderson-Hasselbalch equation, which relates the pH of a buffer solution to the concentration of the acid Ha and its conjugate base A. pH equals pKa plus log A minus HA. pH equals pKa plus log Ha. A minus. Where pKa is the negative logarithm of the acid dissociation constant Ka. A is the concentration of the conjugate base. Ha is the concentration of the weak acid. D. Biological buffers. Blood buffering system. The bicarbonate buffer system HCO is a primary buffer in human blood, maintaining a pH of about 7.4. This system resists changes in blood pH due to metabolic processes that produce acids or bases. Phosphate buffering system. The phosphate buffer system HPO squared is crucial in maintaining the pH of cellular fluids and is effective in the pH range of 6.8 to 7.4. Protein buffers. Proteins, particularly those with histidine residues, act as buffers by accepting or donating protons depending on the pH of their environment. 4. Previous year solved question. Question. Which of the following statements is true regarding weak acids and their dissociation? A. Weak acids completely dissociate in water, producing a large concentration of H ions. B. Weak acids partially dissociate in water, resulting in an equilibrium between the acid and its ions. C. Weak acids have very low Ka values, indicating that they dissociate extensively in water. D. Weak acids cannot form buffers in biological systems. Answer. Correct option. B. Weak acids partially dissociate in water, resulting in an equilibrium between the acid and its ions. Explanation. Weak acids only partially dissociate in water, leading to an equilibrium where the acid HA, hydrogen ions H, and conjugate base A exist in balance. They are characterized by a small Ka value, indicating that they do not dissociate extensively like strong acids. Multiple choice questions. MCQs 1. Which of the following is characteristic of weak acids? A. They completely dissociate in water. B. They partially dissociate in water and establish an equilibrium. C. They increase the pH of the solution significantly. D. They do not release H ions in water. Answer. B. They partially dissociate in water and establish an equilibrium. 2. What is the primary function of buffers in biological systems? A. To increase the concentration of H ions in the solution. B. To resist changes in pH when small amounts of acid or base are added. C. To promote complete dissociation of acids and bases. D. To decrease the concentration of hydroxide ions O. Answer. B. To resist changes in pH when small amounts of acid or base are added. 3. In the Henderson-Hasselbalch equation, what does the ratio A Ha represent? A. The concentration of hydrogen ions in the solution. B. The concentration of the weak acid and its conjugate base. C. The ratio of conjugate base to weak acid in the buffer solution. D. The pH of the solution. Answer. C. The ratio of conjugate base to weak acid in the buffer solution. This concludes the lecture on acids, bases, and buffers, particularly focusing on weak acids and weak bases. These concepts are crucial for understanding the biochemical regulation of pH in living organisms. Let me know if you need further elaboration or additional questions. You said. Equilibrium constant, pH and pH scale and pKa. ChatGPT said. Equilibrium constant, pH, pH scale, and pKa1. Introduction to equilibrium in biochemical reactions. Equilibrium in chemical reactions refers to the state where the forward and reverse reactions occur at the same rate, resulting in constant concentrations of reactants and products over time. In biological systems, equilibrium is essential for maintaining homeostasis, particularly in reactions involving acids, bases, and buffers. The equilibrium constant K quantifies the position of equilibrium and provides insight into the extent of a reaction at equilibrium. The equilibrium constant can be used to describe the dissociation of weak acids and bases, which are central to understanding pH regulation in biological systems. This concept is also closely tied to pH and pKa, which are critical for maintaining the proper functioning of enzymes and cellular processes. 2. Equilibrium constant K and acid-base reactions. A general concept of equilibrium constant K. For any reversible reaction, the equilibrium constant K is a ratio of the concentrations of products to reactants, each raised to the power of their respective coefficients in the balanced chemical equation. The equilibrium constant provides information about the extent of the reaction. If the equilibrium constant is large K and GT, 1, the reaction favors the formation of products. If the equilibrium constant is small K and LT, 1, the reaction favors the reactants. For a general reaction, AA plus BB rightwards harpoon over leftwards harpoon CC plus DD. A plus BB rightwards harpoon over leftwards harpoon CC plus DD. The equilibrium constant, K, is given by K equals CCDD AA BK. K equals CCDD A. K equals CCDD AA BK equals AA. B C D, where C, D, A, and B represent the concentrations of the respective species at equilibrium. B. Acid dissociation constant Ka and base dissociation constant Kb. In the context of acids and bases, the equilibrium constant describes the dissociation of weak acids and bases in water. Ka acid dissociation constant for a weak acid hot dissociating in water. HA rightwards harpoon over leftwards harpoon H plus A minus. Ha rightwards harpoon over leftwards harpoon H plus A minus. The acid dissociation constant Ka is Ka equals H plus A minus HA. Ka equals Ha H plus A minus. A higher Ka value indicates a stronger acid, as it dissociates more in solution. Kb base dissociation constant for a weak base B accepting a proton from water. B plus H2O rightward. The base dissociation constant Kb is Kb equals BH plus OH minus B. Kb equals BH plus O minus. A higher Kb value indicates a stronger base. The relationship between Ka and Kb for conjugate acid-base pairs is given by Ka times Kb equals Kw. Ka times Kb equals Kw. Where Kw. 3. pH and pH scale. A definition of pH. The pH of a solution is a measure of the concentration of hydrogen ions H in a solution. It is defined as the negative logarithm of the hydrogen ion concentration. pH equals minus log H plus. pH equals minus log H plus. Where H plus is the concentration of hydrogen ions in moles per liter m. A lower pH indicates a more acidic solution, higher H concentration, while a higher pH indicates a more basic alkaline solution, lower H concentration. B. pH scale.